Hi, my name is Sarita, lecturer in botany, GDC, W Nalgonda. Today I am going to discuss about structure of anther or microsporangia. This topic belongs to the embryology. Embryology is the one of the unit of BSc second year third semester. Before going to discuss about structure of anther or microsporangia, we have to know about embryology definition. So I want to give brief introduction of embryology. Brief introduction of embryology. The study of embryos and development of embryo is known as embryology. Embryology means the study of embryos and development of embryos is known as embryology. Father of Indian plant embryology is Acharya Panchanan Maheshwari. Father of Indian plant embryology is Acharya Panchanan Maheshwari. Panchanan Maheshwari was a prominent Indian botanist noted for his invention of the technique of test tube fertilization of angiosperms. Panchanan Maheshwari discovered technique of test tube fertilization of angiosperms. This invention has allowed the creation of new hybrid plants. This invention has allowed the creation of new hybrid plants. He also discovered plants according to the difference he found in such embryological studies. So he was called father of Indian plant embryology. Panchanan Maheshwari wrote the book An Introduction to the Embryology of Angiosperms, and he is also editor of Recent Advances in Embryology of Angiosperms. Next, coming to our topic. In angiosperms, the male reproductive structure is called stamens. In angiosperms, the male reproductive structure is called stamens. These are the stamens. In angiosperms, the male reproductive structure is called stamens. The stamens have two parts, mainly two parts. They are filament. This one is filament, green color filament, and anther. Anther is nothing but male reproductive structure. Anther contains male gametes which are called pollen grains. In angiosperms, the male reproductive structure is called stamens. The stamens have mainly two parts. They are filament and anther. This anther is nothing but male reproductive structure. Anther contains male gametes which are called pollen grains. The anther is present. This anther is present at the end of the filament. At the end of the filament, the anther is present at the end of the filament. The anther is two types. One is dithecous anther and second one is monothecous anther. Dithecous anther and monothecous anther. Anther having two lobes is called anther having two lobes. This one is one lobe, this one is one lobe. Anther having two lobes or two thecus is called dithecous anther. It is found in mainly plants in dithecous anther. The septal wall divides the anther into four lobes or four pollen sacs. Four lobes or four pollen sacs. The Anther having only one lobe is called monothecous anther. This is found in the family of Malvaceae. Only one septal wall is found which divides the anther into two lobes, only two lobes or two pollen sacs. Anther. Next. Anther is known as microsporangium. This anther is known as a microsporangium and ovule is known as megasporangium. Anther is known as a microsporangium and ovule is known as a megasporangium. Once again, coming to the male reproductive structure. In angiosperms, the male reproductive structure is called stamens. The male reproductive structure is called stamens. These are the stamens. The stamens have two main parts. The stamens have two main parts. They are, one is filament. This green color is filament and brown color anther. This one is filament and this one is anther. The anther is present. The anther is present at the end of the filament. At the end of the filament. The anther is present at the end of the filament. Thus, anther is nothing but male reproductive structure. This anther contains male gametes which are also called pollen grains. These are the anthers. These are the filament. This is called stamens. Anther is known as microsporangium and ovule is known as megasporangium. Anther is known as a microsporangium. Ovule is known as a megasporangium. This anther is two types. Anther is two types. One is dithecous anther and second one is monothecous anther. Anther having two lobes. This one is one lobe, this one is one lobe. Anther having two lobes is called dithecous anther. It is found in many plants. In dithecous anther, the septal wall divides the anther into four lobes, four pollen sacs. And anther having only one lobe. Anther having only one lobe is called monothecous anther. This is found in the family Malvaceae. Only one septal wall is found in which divides the anther into two lobes, two pollen sacs. This anther is known as a microsporangium and ovule is known as a megasporangium. Next, before going to discuss about structure of anther, we have to know about anther development or microsporangium development. Microsporangium development starts from a mass of primary sporogenous cells. A mass of primary sporogenous cells. This mass of primary sporogenous cells takes periclinal division. Periclinal division means in the group of cells contains, these are the group of cells contains upper surface and lower surface. In this periclinal division takes place parallel to the surface, parallel to the upper and lower surface, is known as periclinal division. A mass of primary sporogenal cells takes periclinal division and forms outer side parietal cells or primary wall layer and inner side spore mother cells. Next, this parietal cells or primary wall layer takes periclinal division and forms anther wall. This anther wall contains epidermis, endothelium, and middle layer tapetum. Next, inner spore mother cells takes meiotic division and produce 4 haploid microspores, which are also known as a pollen grains. Which are also known as a pollen grains. These are the pollen grains. Before going to discuss about development of anther. Before going to discuss about structure of anther, we want to discuss about development of anther. Coming to the development of anther, microsporangia development starts from a mass of primary sporogenous cells. Primary sporogenous cells takes periclinal division. Periclinal division means in the group of a group of initial cells or tissue contain upper surface and lower surface. In this periclinal division takes place parallel to surface. Division takes place parallel to the surface. This division is known as periclinal division. After periclinal division, it forms outer side parietal cells and inner side spore mother cells. Parietal cells are primary wall layer again takes periclinal division and produce anther wall. Anther wall contains epidermis, endothelium and middle layer and tapetum. Inner spore mother cells takes meiotic division and forms 4 haploid microspores or pollen grains. These are the pollen grains. This is about anther development. Next, coming to our topic, structure of anther. Anther wall consists of 4 layers. These are this pink color, epidermis and endothelium. This one is endothelium and middle layers. These are the middle layers and next tapetum. Microsporangium consists of 4 layers. They are this one, epidermis, next endothelium, next middle layers. These are the middle layers and tapetum. Coming to the epidermis. This one is epidermis. It is the outermost layer, 1 cell thick without any intercellular spaces. It is a protective layer. This epidermis is outermost layer, 1 cell thick without any intercellular spaces. It is covered by thick cuticle layer. It is a protective layer. The epidermal cells in the mature anther become stretched and much flattened. Some of the epidermal cells are large in size. These cells are called stomium. The stomium helps in the dehiscence of anther. Stomium helps in the dehiscence of anther. This is about epidermis. Anther, epidermis. Epidermis is outermost layer, 1 cell thick without any intercellular spaces. It is covered by thick cuticle layer. It is a protective layer. The epidermal cells in a mature anther becomes stretched and much flattened. Some of the epidermal cells are large in size. These are large in size. It is called stomium. These are stomium cells. The stomium helps in the dehiscence of anther. This is about epidermis. Next, coming to the endothelium. This is endothelium. Beneath the epidermis, endothelium is present. It is a single layered. It contains single layer. These cells are large in size and do not have intercellular spaces. These cells are made up of cellulose and alpha cellulose. Beneath the epidermis, endothelium is present. It is a single layer. It contains single layer of cells. Below, these cells are large in size and not have intercellular spaces. These cells are made up of cellulose and alpha cellulose. These cells are made up of cellulose and alpha cellulose. Next, coming to the middle layers. Just beneath the endothelium, middle layers are present. This layers is made up of 2 to 4 layers of cells and it stores starch. This middle layer cells are stores starch. This middle layers degenerate by the time of meiosis takes place in the microspore mother cell. Next, coming to the tapetum. Coming to the tapetum. This is the tapetum. The tapetum is the innermost layer of the anther wall. It is surrounded the microsporogenous tissue. These tapetal cells are useful in the nutrition of microspores. These tapetum cells provides the nutrition to developing microspores. These tapetum cells provides nutrients to the developing microspores. The tapetal cells have dense cytoplasm and large nucleus. It based on the development tapetum is two types. Based on the development of tapetum it is two types. They are amoeboid tapetum and secretory or granular tapetum. Coming to the amoeboid tapetum. Amoeboid cells are formed from immature tapetal cells. Example of the amoeboid tapetum: lower dicotyledons and lower monocotyledons. Next, coming to the secretory or granular tapetum. Secretory tapetal cells develops from the mature cells of tapetum. Example: many dicotyledons and higher monocotyledons. Next, coming to the microspore mother cells. These are the microspores. The microspore mother cells undergo meiotic division and gives rise microspores or pollen grain tetrads. The microspore or pollen is uninucleate, single cell and globular. The outer layer of the outer These are the microspores or pollen grains. The pollen grains is uninucleate and globular, single celled. The outer layer of the microspore is made up of pectin and inner layer is made up of cellulose. The outer layer consists of sporopollenin, which checks the natural decay of the pollen grains. It is due to the sporopollenin the pollen grains get preserved during fossilization. The outer layer has pollen kit. The outer layer has pollen kit. It produces a oily coating over some pollen grains called pollen kit. It helps as an insect attract during pollination and protects pollen from UV radiations. Coming to the microspores are pollen grains. The microspore mother cells takes meiotic division and produce microspore or pollen tetrads. The microspore or pollen grains are uninucleate, single cell and globular. The outer layer of the microspore or pollen grain is made up of pectin and inner layer is made up of cellulose. The outer layer consists of a sporopollenin which checks the natural decay of the pollen grains. It is due to the sporolinin that the pollen grains get preserved during fossilization. The outer layer has pollen kit. Pollen kit produce oily moisture. Pollen kit produce oily moisture. It helps an insect attractant during pollination and protects pollen grains from UV radiation. This is about structure of anther or structure of microsporangia. In next video, we can learn about microsporogenesis. Thank you.